Good evening. My name is Daniel, and today I'm going to be presenting on the dopamine D2, D3 receptor agonist Quinpirole and how that gives us a novel possible model for obsessive compulsive disorder. To start out, we're going to talk a little bit about what obsessive compulsive disorder is. So, obsessive compulsive disorder is defined by obsessions, which are going to be followed by compulsions. Obsessions can be anything such as contamination, sensory motor preoccupation, POCD, harm OCD, suicidal ideation OCD. And the compulsions are going to be there to, again, to reduce the anxiety. And common compulsions that we see are hand washing, checking, tapping, isolation, etc. So with this model, we're going to be looking at the corticobasal ganglia circuitry, which is schematized here on the right. And the main thing that's going to be inside of the CBG is the dopamine pathways, which are going to start with their soma inside of the corticobasal ganglia circuitry and then project out into the cortex and other areas of the brain. So our traditional OCD treatments are going to be our SSRIs and SNRIs, which are selective serotonin reuptake inhibitors and selective serotonin and norepinephrine reuptake inhibitors. And then our new OCD treatments that we see nowadays are going to be our antipsychotics, which is similar to what we're going to talk about here. And those are going to be dopamine receptor partial agonists versus the Quinpirole that we're talking about here is a full-on dopamine receptor agonist. So the subjects of this study that we're looking at today were 24 male Lister-hooded rats. It was not stated why they used all male and no female rats. That was just the information that was given in the article that I used for this study. So the weights of the rats were going to be maintained at 95% of their baseline weight through a specific diet of 15 to 20 grams of food per day. The use of the rats was monitored by the UACC, which is the United Kingdom's version of the IACUC that we use here in the United States. And here is just an image of a Lister hooded rat to give you kind of a idea of the subjects being used. So the actual article that I used for this project had multiple experiments within it. And if I was going to present on all of those, it would take quite a long time. So we're just going to look at the main experiment that was in the article, and that is the effects of Quinpirole on checking-like behavior in the Lister-hooded rats. So in this experiment, our methodology was to give the Lister-hooded rats the dosage that you see in the first bullet point of Quinpirole for 10 consecutive days, and this was going to be administered in a room that was not used for behavioral analysis. After they were given the injection of Quinpirole, they were placed into separate holding cages. And on days 1 through 3, they were in these holding cages for 60 minutes. And then on days 4 through 10, they were only placed in there for 20 minutes. The reason behind this is to monitor for any post-administration side effects. And on days 4 through 10, this was reduced because the rats had gained a tolerance to the Quinpirole and were not showing as many of the post-administration side effects as they were on days 1 through 3. The testing portion of each trial of the rats lasted about 21 minutes, and this consists of being placed in a box with observing levers and active levers. A little bit more about that. Observing levers are going to be a lever that, when pressed, will illuminate a light in the box somewhere, which will indicate the location of an active lever. An active lever is going to be a lever that, when pressed by the rat, is going to release a treat. And so how this maps over to the human-like model of obsessive compulsive disorder is the observing levers are going to be our checking or compulsive-like behaviors, and the active levers are going to be our proper coping techniques with OCD, such as cognitive behavioral therapy, cognitive reprocessing or restructuring, I should add. And so that's how we map this over. The results of the study itself showed that after a daily administration of Quinpirole, the DA receptor agonist, the rats showed an increased observing lever presses. They also showed an increase in extra observing lever presses. Now, the extra observing lever presses is not an entirely different lever in and of itself. It basically just means that the rats would return to the observing levers and press them repeatedly. This also showed that reduced active and inactive lever presses decrease the total amount of treats that the rats would earn throughout each of the trials. So what does this mean? Basically, it means that quinpirole increasing observing lever presses, indicating OCD-like checking behavior or compulsive behavior, according to the Schützman and colleagues' animal model of OCD that was developed in the 80s or 90s. So it's a relatively old model. Even after the administration of quinpirole was ceased, we also saw that this checking or obsessive like behavior or compulsive like behavior would last for 2 to 3 weeks, indicating that the areas of the brain that are responsible for this checking like behavior are a neural circuitry or neural pathway that must undergo a cognitive restructuring through neuroplasticity. And in humans, we see this as our cognitive behavioral therapy, which allows us to restructure our neural pathways in order to handle the intrusive thoughts and decrease our compulsive like behavior. We also saw that the checking behavior, so the observing lever presses and extra observing lever presses, did not increase the rat's ability to find the active levers and gain that treat or that reward. And this maps over into the human model of obsessive compulsive disorder, showing that the checking or compulsive like behaviors do not actually increase the success rate of reducing anxiety. In fact, we see in the human model of obsessive compulsive disorder or in patients who have OCD that this compulsive like behavior will reduce the anxiety temporarily, but that anxiety will return when the intrusive thoughts return because these compulsive like behaviors are not a viable source of anxiety reduction. Instead, the best treatment for that as of now would be our SSRIs, or in our newer model or in our newer treatments, we see these antipsychotics or cognitive behavioral therapy. And obviously, the best one is going to be a combination of both medication and therapy. So, what does this tell us, and what can we use this information for? Well, basically, the main thing is that this study allows us to imply that a possible treatment for OCD would be a dopamine D2 D3 receptor antagonist. And this would decrease these checking like behaviors since the agonists, which are going to increase the affinity of dopamine to the receptors, uh, if you give an antagonist, it's going to decrease this affinity and therefore possibly decrease these checking like behaviors. Now, we see in these antipsychotics, which are partial agonists, which allow for partial increase in affinity of the dopamine to the receptors, that there is a decrease in our compulsive or checking like behaviors. So there is a possibility that we could use a slightly less potent agonist or partial agonist in order to achieve this reduction in checking like behavior, or a full on antagonist, which would possibly have greater side effects than using just a less potent partial agonist that would be less potent, again, than our antipsychotic medications. The study also implies, as we saw in the last slide, this latency period between onset of treatment um, and the onset of reduced compulsive behaviors, which I think I meant to say here, the actual um, ending of treatment and the reduction of compulsive behaviors. So, and that's similar to what we see in patients with obsessive compulsive disorder, is that it does take a while after starting cognitive behavioral therapy or starting medications. So, how that would map over to this study would be just completely stopping the quinpirole administration. But it does take a while after starting CBT or medication to see this improvement in our checking like behaviors or this reduction in our checking like or compulsive behaviors due to this neuroplasticity that is responsible for restructuring those neural pathways that are involved in the checking like behaviors of obsessive compulsive disorder. So, one thing again that I would like to see is this possible medication of the antagonist being used in a future study in possibly Lister hooded rats and see how that affects the checking like behaviors and seeing if we could map that over into human models to completely reduce or even eliminate the checking like or compulsive behaviors in OCD. Here is the citation that I used for this project. This is the actual journal article. It is a peer-reviewed journal article. I did find this very interesting, and I really enjoyed researching this as someone who has obsessive compulsive disorder and wants to go into a neuropharmacology program to study more about the effects of neurotransmitters and other brain like hormones and molecules in and how they are responsible for mental disorders, especially in OCD. Thank you very much, and I hope you enjoyed the talk.